21st 21st 2022 today 21st 2022 MLT second year. Subject is microbiology. Topic is yesterday we are discussing Candida albicans. Today we are discussing in Cryptococcus neoformans. Cryptococcus is a substance or chronic infection caused by yeast. Cryptococcus is caused by Cryptococcus neoformans. Cryptococcus neoformans. It is a yeast. Morphology. The capsule characteristic of this species contained by microfibrils that prepared or radiate from the cell wall to coil and intertwine in various directions and uniformly electric bases present of complex polysaccharide molecules. Then Cryptococcus neoformis is a round budding cell 4 to 20 micrometers in diameter. Micrometers in diameter. It is a polysaccharide capsule. It is a polysaccharide capsule. And Cryptococcus neoformans is a infection occurs. How to infected means inhalation. Inhalation means mringadam by through the air or water. Cryptococcus neoformans infect the lungs, causes pneumonitis. Cryptococcus neoformans causes pneumonitis. Pneumonitis, inflammation of the lungs is called pneumonitis. The next point, cutaneous lesions. Cutaneous lesions from the small ulcer organelles. Cryptococcus neoformans, some patients are seen in cutaneous lesions or small ulcers or granules. Visceral lesions stimulate tuberculosis and cancer. Some patients are seen visceral lesions stimulate to the tuberculosis, TB, or in cancer. Cryptococcus meningitis, the most serious type of infection. Cryptococcus meningitis is a serious type of infection. The onset is insidious and slow. Cultural characteristics. Cryptococcus neoformans reproduces by budding and forms round yeast-like cells 3 to 6 micrometers in diameter. Some Cryptococcus neoformans cultural characters you have to see is especially reproduced by budding and forms round yeast-like cells are seen. Sometimes Cryptococcus neoformans are smooth, convex, yellow, or tan colonies or solid media. Tan colonies are solid media. And especially Cryptococcus neoformans, they are growing in the room temperature 26 to 28 degrees centigrade. Most non-pathogenic Cryptococcus strains. Some Cryptococcus neoformans consist of two strains, pathogenic and non-pathogenic. Pathogen means causes for the disease, non-pathogen means is not causes for the diseases. So, especially non-pathogenic strains are in a Cryptococcus strains, especially they are growing in the 37 degree centigrade. Pathogenic strains are growing in the 28 degree centigrade in room temperature. Cryptococcus neoformans, lactose and nitrates are produced as pseudomycelia and cornmeal rice trine agar. Most strains Cryptococcus neoformans especially used for creatinine as a nitrogen source, which another useful of biochemical reactions in Cryptococcus neoformans, especially their non-pathogenic strains to distinguish produce melanin. And fungal enzyme phenol oxidase acts on a certain substrate. Example, dehydroxyphenylalanine. Next heading, lab diagnosis. Demonstration of capsulated budding yeast cells in lesions and biculture. You have to do in separate dextrose agar on a culture medium the growing of Cryptococcus neoformans. How to they are how to growing you have to what is the colonies are seen in a budding yeast cells in a lesions. So the capsules are prominent Indian ink preparation. This is the laboratory test. How to you have to done with the Indian ink preparation. You have to see in microscopic examination. So culture it grows readily separate dextrose agar is a fungal culture media. Uh, Next point, what is the growing is a especially their non-pathogenic strains are growing in 37 degrees centigrade. Pathogenic strains are growing in the 28 degrees centigrade only. So, Lab D in diagnosis of Cryptococcus neoformans infection microscopically, which samples you have to collecting. Example, one is sputum, one is fluid, cerebrospinal fluid. Cerebrospinal fluid and sputum, you have to growing the Cryptococcus neoformans. Why? Because there are Cryptococcus neoformans presence in the cerebrospinal fluid and sputum sample. Next point, uh, especially smooth and mucoid kinase. In culture, several dextrose agar, smooth mucoid cream colored colonies are seen. Smooth mucoid color colonies are seen in a culture media on separate dextrose agar. Next, cryptococcal fungal culture as essentially Cryptococcus neoformans cannot currently distinguish from the C. gitti, Cryptococcus neoformans, gitti. So, Cryptococcus is one of the most common life-threatening fungal infections people with AIDS. Cryptococcus neoformans, very important STD disease, a sexually transmitted disease. Example, HIV patients. Especially, we have to see in HIV patients, you have to take the sputum sample, isolate from the Cryptococcus neoformans. Why? Because the sexually transmitted disease person consists of Cryptococcus neoformans. That's why they are growing in the separate dextrose agar. In colonies are seen in smooth and cream colored colonies are seen. These colonies are Cryptococcus neoformans. So, especially in normal patients and sexually transmitted patients, both the patients in consist of a Cryptococcus neoformans. Cryptococcus neoformans causes for pneumonitis. Pneumonitis, inflammation of the lungs is called pneumonitis. Underline inverted commas. So, next heading, next topic is a dermatophytes. Definition: dermatophytes a group of morphological or physiologically related modes. Important point, underline inverted commas. Dermatophytes as a morphologically or physiologically is a modes. So next point, dermatophytes phytosis. Cutaneous or ringworm. Cutaneous or ringworm. Continuation tomorrow. Say thank you all of you.